Hello, my name is Amanda Adolfo, and I will be presenting a quick lesson on fret microscopy, which is a topic that we learned about in MCDB 165A. So what is fret microscopy? It stands for fluorescence resonance energy transfer. From the name, you know you're going to utilize fluorescence microscopy. However, the resolution of a fluorescence microscope is several hundred times less than the size of a typical protein. So how are we able to determine if proteins are close to each other? Here's an analogy that I found on the Nikon Microscopy U page. So conventional wide-field microscopy can tell us that two students are both present in a large lecture hall, CS50. However, the microscope can't tell us if they're in the same discussion classroom or if they're sitting in adjacent desks. To solve this problem, fret microscopy allows us to see a more precise localization of two fluorescently labeled proteins and even their interaction. We will see how this is all possible further into the presentation. Some tools that you will need to use fret are a fluorescence microscope, which could be wide field or a confocal microscope. Obtain a fixed or living sample that you are able to fluorescently label. Most commonly, you can have the protein express the colored label. And lastly, you will need to choose individual fluorophores that have separated excitation and emission spectra. One important thing to note is that the chosen labeled molecules have to be very close in order for fret emission to occur. So this has to be within 10 nanometers or less. So how does this all work? As mentioned before, having separated excitation and emission spectra is important when choosing your individual fluorophores. It's important to have this distinction so that you can see the labels better under the microscope, and you can also see when fret is occurring. It minimizes a form of noise called crosstalk, which we will talk about later in the presentation. Since you will choose two different fluorophores, you will also need specialized fluorescence filters for the microscope that can help you examine the proximity of the labeled molecules in the sample. You will need a donor emission filter and an acceptor or fret emission filter to visualize these labels. So now we'll talk about the specific mechanism that makes fret work. First, you will excite the donor fluorophore at a specific wavelength using the light source on the microscope. If the acceptor fluorophore is within range, the donor fluorophore can transfer its excited state energy directly to the acceptor without emitting a photon. The acceptor molecule quenches or diminishes the donor molecule fluorescence, while there is also an increase in acceptor fluorescence emission. We can break this down a little further and take a closer look at resonance energy transfer. So we are looking at a Jablonski diagram, which shows the coupled transitions between the donor emission and acceptor absorbance in fret. The absorption transitions are represented by the green arrows, and the emission transitions are represented by the red arrows. You can pay special attention to the dotted arrows, where the donor fluorophore transfers its excited state energy directly to the acceptor without emitting a photon. The resulting sensitized fluorescence emission has characteristics similar to the emission spectrum of the acceptor. The method that we just described is called sensitized emission, where two-channel imaging or two-color ratio imaging occurs. So just to reiterate, the donor fluorophore is excited by a specific wavelength from the light source, and the signal is collected using emission filters chosen for donor fluorescence and acceptor fluorescence. A measurement of fret can be recorded by taking the ratio of acceptor emission intensity to donor emission intensity. This can be at one time point or over time, depending on your experiment. Sensitized emission is one of the simplest methods of fret. However, there could be crosstalk between fluorescent proteins, which could pose potential problems. Crosstalk is the presence of bleed-through of the background signal or cross-excitation of spectrally overlapping fluorophores. In order to minimize these issues, we can do a few things. For one, we can test several different filter combinations across many samples. So we can do the following controls, such as using samples with only the donor, samples with only the acceptor, or look into samples that have our proteins of interest that include both the donor and the acceptor. As mentioned before, that's why it's important to choose fluorophores that are well separated spectrally in order to reduce cross-excitation. By doing these corrective approaches, we can minimize crosstalk. So now we can talk about different ways of using FRET through fluorescent protein biosensors. These are great for giving us information about intracellular processes. The first fluorescent protein biosensor is a calcium indicator called Chameleon, and it is made by sandwiching the protein calmodulin with the calcium calmodulin binding domain of myosin light chain kinase, or M13 domain, and these are put between blue and green fluorescent proteins. In the presence of increasing levels of intracellular calcium, M13 domain binds the calmodulin peptide to produce an increase in FRET between the fluorescent proteins. Calcium acts as a second messenger in several signaling pathways of the cell, so this is a good method to use for that. Secondly, we have phosphorylation biosensors, which consists of a peptide that has a phosphorylation motif from a specific kinase and a binding domain for a phosphopeptide. This pair is sandwiched between two fluorescent proteins. When the biosensor is phosphorylated by the kinase, the phosphopeptide binding domain binds to the phosphorylated sequence, thus activating or diminishing FRET. This method can generate highly specific biosensors. The last method on our slide is the protease cleavage assay. Two fluorescent proteins are linked together by a short peptide sequence that contains a protease cleavage site. This sensor has a strong energy transfer that is destroyed when the linker peptide sequence is cleaved. The largest family of protease biosensors incorporates a cleavage site to one of the caspase family of proteases, which allows the sensor to be visualized during induction of apoptosis. On this slide, we can look at an example of FRET in action. As you can see, a yellow fluorescent protein and a cyan fluorescent protein were linked together by a short peptide linker sequence, where FRET emission is occurring. Upon the introduction of a protein, the protease, the sequence was cleaved, resulting in no FRET and only donor CFP emission. So if you remember from the previous slide about what this method is called, it's a protease cleavage assay. Here's another example of a living cell culture that is looking at proteins involved with apoptosis. So in this case, the donor protein is labeled with blue fluorescent protein, and the acceptor protein is labeled with green fluorescent protein. These proteins were imaged in living cell culture, and they are located in the mitochondria. From these images, you can see that they display a gradual decrease in binding as programmed cell death proceeds, or apoptosis. This image provides, this microscopic image shows that the picture on the left is donor emission, or BFP. In the middle, it's only acceptor emission, or GFP, and then the FRET emission is imaged on the right. So it's important to note that they use separate filters for all of these images. So here's a quick review of FRET. So you can use fluorescence microscopy, either wide field or confocal, to visualize FRET. And FRET is beneficial because you can use it with a fixed or living sample, and you can have special emission filters. You can choose a donor-acceptor fluorophores that have separated emission spectra in order to reduce crossover or crosstalk. FRET occurs when molecules are very close, within 10 nanometers, so that the excitation energy from the donor fluorophore can transfer to the acceptor fluorophore, which results in FRET emission. And lastly, fluorescent protein biosensors can be used for reporting intracellular processes. And for fret measurements can be taken by obtaining the ratio of acceptor emission to donor emission. So the protein biosensors that we talked about were chameleon, which is activated upon increased calcium presence, phosphorylation biosensors, so when the phosphorylation state of the kinase depends on whether fret is activated or destroyed, and lastly the protease cleavage assay, where when the linker sequence is cleaved, the fret signal disappears. So thank you for coming to today's lesson about fret, and here are my references.